Okay, I want to begin by thanking Phil. And I want to today, in the next 10 minutes, sort of highlight some of the unique challenges that people in the kinase field are facing, especially with all the clinical data coming out on these proteins. And we've already heard about the importance of these proteins in cell cycle regulation. And if we're going to be thinking about 3D virtual modeling, and these proteins pretty much control, 30% of the cellular processes, we really need to be thinking about these proteins in greater detail. So what I'm going to do is essentially tell you some of the challenges we in the field are facing, and then how we're beginning to address some of these challenges using some semantic web technologies. And if I have some time, I'm going to briefly comment on some of the educational efforts using these tools. So. Okay, so again, Susan is going to highlight the complexity of the system a little bit more in her talk, but I just want to briefly comment on the kinds of complexity that we're dealing with. So we have structures of many of these kinases, and what I've highlighted here is the prototypic kinase pKa that's been very well studied. But despite all the information that we have, we still don't understand quite a bit about these proteins, largely because of the complexity. At the protein level, there's this complex network, communication networks that we still don't understand how they allosterically communicate between different parts of the molecule. There's complexity at the dynamic level and the assemblies, and Susan's going to highlight that, so I'm not going to touch upon that. And obviously, there's complexity at the cellular level and at the tissue level and at the organism level, as Peter pointed out. So how are we going to begin to address all this complexity? And I'm going to sort of take a more data-centric approach. And how can we use existing data to begin to address and appreciate the complexities that we're facing here? So thankfully, these are very well-studied systems, so we have a wealth of information here. And Phil just walked in, so I'm going to, highlight the importance of integrating all this data, which I think most of the community already appreciates, but sort of emphasize the importance of evolutionary data. I mean, that's Cell's own experiments, and yet we haven't really in this meeting, thought about, incorporating that data in understanding these complex systems. So this might be maybe a topic of interest or a topic of another meeting. So we have all the evolutionary data, and we're actually spending a lot of time thinking about what evolution is telling us about these complex systems and how we can begin to model these systems using evolutionary information. But the challenge, as you can see here, is even though we have all of this information, the challenge is data integration. And so how do we really begin to address this challenge? So I'm going to use this simple query, and this, again, relates to Phil's comment earlier yesterday as to the scientific inefficiency. And people in my group, when they come in and they want to test the impact of, say, a given cancer mutation in a given kinase, they essentially do the same thing of, they gather mutations from Cosmic, map them onto the PDB, and then look at what subdomains they map to. And then everyone who wants to do this kind of, iterates the same process over and over again. So how can we really come up with a conceptual framework where we can kind of do this in a way that you don't need repetition? And if you want to, formulate aggregate queries, and this is, people in the community want to do this sort of queries in a daily basis, and yet they don't have a framework for doing this. For example, what are the pathways that are targeted by mutated kinases, and what kinase subgroups the kinases, the mutated kinases, fall into? I mean, you essentially have to write, customized scripts or programs to do this. So in the past 2 years, we've been thinking about how to really conceptualize this knowledge and how to address this knowledge, this challenge that we, particularly in the lab, are facing, and presumably the community itself is facing, by conceptualizing all of this in the form of an ontology. And I'm sure many of you in this room already know what ontologies are, but for those of you who do not, this is essentially a way of conceptualizing data in ways that humans can read, and both computers and humans can read and understand. So here's a simple schematic showing how we in the field like to think about all of these disparate sources of data. So I typically, if you want to think about a gene, you want to think about what disease it's implicated in, and the semantics that's indicated here is pretty much the same language that we in the community use. So this could also provide a framework for integrating communities. And so if you want to describe a mutation, you want to see whether the gene has a mutation, and if so, if it has a mutation, which subdomain it's located in. And again, subdomain is a concept that people in the community, the protein kinase community, use a lot. But and by introducing these semantics, we're able to it sort of provides a framework for integrating communities as well. And you want to see, what gene, what pathways a gene is, participating in. So by conceptualizing data in the same way as we in the community sort of talk about data, I think we have a way of both forming a community, sharing data between communities, and also doing data analysis in ways that we can never achieve through other resources. So we have actually built a focused ontology on the kinases, which captures information on the sequence structure, function, and regulation. This is sort of a simple schematic or data model of how we're capturing this. And this is a workflow of how we're starting from these disparate sources and then mapping them and then populating the ontology. And I'm going to very briefly tell you about some of the applications of how we're using the ontology to perform systems level analysis of the data. Before I do that, is just a snapshot of the viewer, so anybody who's interested can essentially navigate all information related to any kinase, the disease, the relevance, the functional aspects, regulation, all of that in one place, and that could be a way to kind of navigate everything in one place. So it facilitates data integration. And so here's getting back to that same question that I previously posed to you. What is the challenge? So for example, as I said, one of the common queries that people would like to ask, what cancer, what subdomains are the mutations mapping to? This is a common query, and we can kind of rapidly do this because we've conceptualized the data using pretty much the same semantic, terminology. So for example, mutations in subdomains or classes, and located in is a property that relates these two classes. So you can pretty rapidly see, what subdomains these are mapping to. So if you tell a kinase researcher that a mutation is located in subdomain 7, that's already giving them more information than just saying a mutation is located, within this gene at position 384. So it immediately rings a bell, and they can then begin to think about the mutation in the context of the structure, even though they're not really looking at a structure. So again, this is another query where you can see how pathways, mutated kinases participate in various pathways. And when you do these sorts of queries, you can see that there are some pathways that are popping up that previously people really didn't appreciate were implicated in cancers, like for example exon guidance. And you could do these sorts of queries on a cancer specific. So you could look at specific cancer types and see what pathways they're targeting, and that can then provide a framework for formulating hypotheses. So I'm going to, in the next couple of slides, very quickly show you how we're using the ontology framework for reasoning and hypothesis generation, using a couple of slides. So here's a simple query. You want to see, out of the 20 amino acids, which amino acids are more frequently mutated in cancers. And so this is a very simple query that you would like to see. And until now, nobody had really actually performed a query like this. And you can see that arginines are more somehow more frequently mutated. And I certainly didn't expect this until I really saw this data. So with this data, then you can formulate another query. Okay, what structural regions are these mutations mapping to? And obviously, you see what you would expect to see. There are some regions that these arginines are mapping to. And once you see their mapping, you see that some the arginines that are mutated, in fact, are coordinating with the phosphate group. So this is again linking to the structural data and linking to the concept of how the devil is in the details and integrating using a top-down approach. You can, with this data, now begin to formulate a specific hypothesis as to how mutation of that arginine would then, contribute to abnormal regulation of a kinase. So in summary, I'm going to sort of conclude by saying that, investigating complex systems obviously needs numerous hypotheses to be formulated and tested, and ontologies provide a framework for accomplishing this goal. And I didn't get a chance to, talk about this, but I think, by building focused ontologies, we could also have a framework for just integrating communities, because there are certain semantics and terminologies that people within a particular community use, and that could really nicely provide a framework for integrating communities. And I haven't really, touched upon this issue, but we're also beginning to use this as a framework for teaching students how integrate, how multiple or distinct concepts in biology can be integrated using the same semantic languages that experts in the field use, using ontologies as a framework. So with that, I want to thank, and I have to tell you that much of my lab really focuses on thinking about the evolution of these systems, and we've only recently begun to use ontologies as a framework for integrating data, and much of this is due collaboration with Chris at Kosher, who's a semantic web expert at the CS department. And I want to thank funding from the National Science Foundation and the Cancer Society, Georgia Cancer Coalition, and the University of Georgia. And thank you for your attention.